Would you please introduce yourself? Tell us who you are.
Sure. I'm David Goldman. I'm chief of the Lab of Neurogenetics and clinical director of NHAA, which is the Intramural Alcohol Research Institute program.
Yeah, thank you for talking with us today.
Sure, Graeme.
So, how did you move along your alcohol research? And you could go way back. Were there influences in maybe your childhood, your adolescence, high school? Were there people that had a special impact on you?
Sure. I'm from a medical family. Father is a pediatric immunologist. He died a few years ago. And there are other physicians in my family. So a bit of a science tradition. When I was in high school, even I had been in labs. Then I was lucky that when I was in medical school, I was also able to be in a few labs. And, these were neuroscience labs, but also another lab that did immunology. So I actually had a lot of lab experience before I ever went off to university. And went to Yale, graduated in 3 years. When I was in my second year, I figured out that I really didn't have enough money. So what I did was, and I know your connection to Yale, Graeme, so this will amuse you.
I am smiling. Yeah.
Right. And so I realized that I could graduate in 3 years, and then applied to the University of Texas medical schools, because you would do one application and apply to all these medical schools. So they accepted me, and then I went to the medical school in my hometown. When I was a senior medical student, I was able to do a year of research, and 3 months of that was an externship at NIMH. So I came to NIMH, did research on non-human primates as just as a student fellow, and made some contacts there. And I was lucky to be able to be offered a job. And after another year of residency in Galveston, then I actually came to NIMH and NHAA. I was the first intramural NHAA fellow. People think that these institutes have been around forever, but of course they haven't. The NHAA was formed in the late 1970s. And first it didn't have any program or grants, and then it didn't have an intramural program. And so initially, all they could do was to put me in an NIMH lab, but this is the National Institute of Mental Health course. And that was a great lab. It was the lab of Giulio Cantoni, who studied and discovered S-adenosylmethionine. But more importantly, my mentor was a guy named Carl Merrill, who's a very creative type. He was doing something called two-dimensional gel electrophoresis. It was a early proteomic method, one of the first omic methods. It let one quantitate and actually qualitatively and quantitatively study up to about 1,000 proteins in a cell at a time. So we did a lot of studies, some which were on genetic diseases, others which were on what you call conditions. And as I was doing that work, I became a geneticist in a couple of different ways. First, because I was discovering genetic variation in the course of that science, and also because one of my early collaborators was Steve O'Brien, who's an evolutionary geneticist at NCI. So, this brought me into the genetics field. I began teaching a graduate course in human genetics at a local university, George Washington University. And that's where I really learned genetics, because I'd never taken a genetics course. But, as you teach it, as you do the science, then you, of course, have to learn. Um, when when I really began to do any research on alcohol, I think it was partly because I met two people. One was Mark Shuckett, and this was at a time that Mark was first discovering that some people are more sensitive to alcohol than others, and they're less likely to become alcoholic, as we would call them then. But now we would say less likely to have alcohol use disorder. And then also Richard Dietrich, who on the rodent side was studying mice that are kind of the equivalent of the alcohol um, sensitive or insensitive people. These were the long sleep, short sleep mice. This was all during an era where it was commonplace in the animal research to select for strains that were high and low for alcohol sensitivity or preference or withdrawal. And so I got to know many of the people who were doing that, but then also to do genetics with Richard Dietrich, who was a big guru in the field, and at University of Colorado was a big advance for me. And also I did personally meet Mark Shuckett and learn about his studies on human alcohol sensitivity. So that was back in the early 1980s, actually. So in my own career course, I was first a clinical associate in NIMH. And that was after really just a year of being this Alcohol Institute fellow. But then in around 1984, 85, I got offered the opportunity to come to NIAAA when NIAAA founded its first intramural lab. And that first intramural lab was run by a Finnish scientist named Markku Leinola. And so in that lab, I set up a small genetics unit. We're doing first the proteomics, but immediately I saw the power of the nucleic acid based studies, the DNA studies and studies on RNA, because those were studies where you could rapidly move to the identification of that which you were studying, and also because a new method called PCR had been invented around then by Kary Mullis. It had actually been discovered conceptually more than a decade ago by the Nobel laureate Khorana. But in this method, one could target a particular DNA sequence, amplify and purify it, and then via sequencing, identify or verify what you had amplified for your qualitative or quantitative study. The quantitative studies being based upon difference in sequence, and the quantitative studies being based on the amount of the sequence that was present. So I was jumping into a field which had been really dominated for a couple of decades on the animal side by these types of artificial selection experiments where you select an animal to be high or low for a trait. And then the human side had been dominated by first the twin paradigm to establish heritability, which established that alcoholism was moderately to highly heritable. And then on the gene identification side, by two different paradigms. The first paradigm being the family linkage study. And family linkage studies were all the thing back in the 1980s. And then secondly, by the candidate gene approach, where by understanding pathways and the way that alcohol was metabolized or might work, that if you would study the genes for that, that you could then unravel differences in vulnerability to alcohol use disorder or to response to alcohol. And that was actually, both of those were fruitful approaches. We, in our linkage studies, decided that we would focus on population isolates. So I initiated studies in Native American populations, initially doing a kind of grand tour through the Southwest to identify um, um, from prospective sites that we might do such studies, actual sites where we could carry these out, and eventually did studies which were at the DNA level and also the deep psychiatric and neuropsychological phenotype level on tribes in Oklahoma, Plains Indian tribes, and also some Southwestern tribes. So as we did that, we were actually also following the footsteps of work that had been done by Spiro Manson and Jim Shore, again at Colorado. And what Manson and Shore had no connection really to the alcohol field had done is that they had been able to find that in Native Americans that alcohol use was was damaging, and that alcohol use disorder itself was similar in its structure or phenotype as alcohol use disorder in other populations, and that was really important. We expanded on that. We showed that if you had either AUD or if you were a binge drinker, that if you were Native American, you were worse off in almost every area of the DSM or in every area of the DSM. And that was a different perspective than what some anthropologists, such as Phil May, were finding. They had actually been proposing that AUD might be the reification of a culturally normative behavior, that it was in some way normal for Indians to drink a lot, and that which ran counter actually to what a lot of the tribes believed. The tribes noticing that AUD was their number one health problem, responsible for social dislocation, unemployment, abuse, everything else that tracks with alcohol use disorder. So we did observe the same thing. Some of the papers that we published were epidemiologic and phenotypic in their sort of nature, showing these disastrous effects of alcohol, showing how alcohol use disorder was often preceded also by early life stress, including child abuse. So some of this work we carried out with leading experts on child abuse trauma, such as Mary Koss, who did the big study for the CDC on abuse and trauma. But of course, it was a genetic study, so we were doing linkage studies in the families that we collected. And we were also studying some of the candidate genes. In the linkage studies, we were able to, along with another group that had just started to do their science at around the same time, POGA, to identify in our families that there was a gene or genes on chromosome 4 near the alcohol dehydrogenase gene cluster that contributed to AUDs. That was the major finding of our paper, which was an early paper in American Journal of Human Genetics. And in the same issue was a paper from the KOGA group, which in non-Native American populations, cosmopolitan population of the United States, also implicate the same region of chromosome 4. And what's interesting is that to this day, in the genomic studies that are done, still the two loci that, or genes that exert the strongest effects on risk are the alcohol dehydrogenase gene or gene cluster on chromosome 4 and the aldehyde dehydrogenase gene. And so these are two genes that are involved in consecutive steps of alcohol metabolism, and they have common variants, especially common in East Asians, but also certain other populations that disturb the rate of metabolism, leading to the so-called East Asian flush, which discourages drinking. But unfortunately, if you do drink and have this variant that makes it more difficult to metabolize acetaldehyde, or if you have the ADH variant that leads to more rapid production of acetaldehyde, then there's a risk of upper GI cancer, breast cancer, other cancers that are triggered by the carcinogenic potential of acetaldehyde, which is a mutagen that's recognized by the International Commission.
May I pause it for a moment?
Absolutely.
Well, first of all, one thing.
And thank you for interrupting, because I'll just go on.
Well, I'm enjoying this history. One thing that I like about this history so far is that you're mentioning things that, since I arrived in the alcohol field later, I accept as if they were given to us by Moses. But clearly, nothing is.
And they're not. And they're not. It's
they had
all
to be learned.
They really had to be learned. For example, because it's got to be understood that the human genome sequence was only in 2001. And prior to that, if there was a gene that was isolated and cloned, it was studied at the protein level, and then it had to be cloned at the DNA level. So, for example, my lab cloned one of the alcohol dehydrogenases. It's actually an obscure aviation, so I won't really go on about it, but it's the class 5 ADH, which is also the formaldehyde dehydrogenase. But meanwhile, for the ADHs, you had people like Akira Yoshida, who cloned, and T.K. Lee, who were cloning ADHs, ALDHs, and nobody recognizes that anymore, because now you just have the genome sequence, and there the genes are there. But to do that, the proteins had to be purified, peptides had to be made from them, those peptides sequence, and then DNA library screened with little probes, and then you pull out the bacterium that's expressing that gene that your probe recognizes, and purify from that bacterium the gene that you were trying to clone, sequence it, and that's how it was done, one by one, instead of everything all at once. But yeah, you're absolutely right. We stand apart from that much earlier biology, because now that's a history that existed before 2000, but also the modern methods are so much more powerful, as we'll be talking about.
I'd like to ask you for one detail level, but would you please explain what a linkage study is?
Yeah, so in a linkage study, what you do is that you look for how either the locus, which means the gene, or the allele, which means the variant of the gene, tracks with another phenotype. Or it could be how one gene tracks with another gene, which is how the genetic maps were built. And so family linkage is locus-based linkage. You have a variant which can be in coupling or in repulsion with the traits. You might notice that in families, that hair color predicts either whether that you are
So for those of us who are not integrated, currently working with genetic studies. Would you please explain what a linkage study is?
I'll try. So a linkage study is that we try to relate a gene to a gene on a genetic map or identify a gene that might impact a phenotype or a trait. And there really are two ways of doing linkage. One is linkage by allele, where you identify the actual causative variant. And so you might be identifying, for example, that the beta globin variant, allele 6 val, that causes sickle cell anemia, causes sickle cell anemia. So that is linkage. But also, one can do allele-based association at a gene where it's not the actual functional locus, like this allele 6 valine amino acid substitution. It might be a nearby variant, and that depends upon a phenomenon called linkage disequilibrium, where in the population, one marker tends to travel with another. So that's a way of doing linkage. The way of doing linkage that really was popular in an earlier era, and then we're coming back to a little, was family-based linkage, which depends upon another phenomenon called meiotic recombination. So whenever the germ cells are made, the sperm and the eggs, our genetic material is reshuffled and recombined. And there are anywhere from 1 to 4 of these recombination events in a chromosome. And so when that reshuffling occurs, then the old genetic recombinations, the old genetic combinations on a chromosome are disrupted and inherited independently. And as Mendel actually originally proposed. So what one has is then linkage maps based upon recombination and recombination distances. What is the likelihood of recombination? So this type of linkage can implicate and can cover much larger regions of the genome because recombination occurs over millions of base pairs, whereas the linkage disequilibrium is over small regions. Also, though, this linkage in family, this locus-based linkage, is very different in terms of the fact that genetic markers can be in coupling or in repulsion with the trait that you're interested in. So you can show that the gene is linked by virtue of a marker at the gene. And in one family, the one form of marker or allele might be linked, and then in another family, the opposite form might be linked. And so, we were able to take that into account. It really was after World War II with Newton Morton that the whole linkage revolution began in families. And then it was after the genome revolution with the sequencing of the genome, discovery of some 22 million common polymorphisms, that the linkage disequilibrium or association method became so powerful. And that was in about the year 2000.
Thank you.
You're welcome. Are we recording still?
Sure. Yep, the end button is shown.
Excellent. How can I tell, actually?
There's the end there.
End. Okay, that's good. If that should ever disappear, I'll let you know immediately.
I'm also watching the solid green light on your microphone. That tells me if that antenna has dropped.
Excellent.
Okay. Now, that was what we just covered. That was all part of your answer from the initial question. My initial question, I think, we talked a bit about history of genetics. So had you already talked about? You already talked about how you got your path.
Don't worry if it's duplicated.
Yeah. I think what challenges have you faced along your path? And you did mention that you were running low on money, so you finished college in 3 years instead of 4.
Right. Ran low on money. Also, as a young investigator, I was helped greatly by the ability to get actually HIV money to support our studies on population isolates, mainly the studies in Native Americans. And, in that regard, really helped by Boris Tabikoff, who was scientific director of NAAA at that time. Also, just as a young investigator, you have a small group. And so as a young investigator with a small group, putting gambles or you're putting your chips on certain scientific ideas. And some of these ideas pay off and some of them don't. Sometimes your idea is not such a wise choice. Actually, early in my career, a number of these things that I worked on were from findings that others had published, which turned out to be false leads or red herrings. I'm sure that you're familiar with that idea, but there are papers in Science and American Journal of Human Genetics, and they provided important leads that we wanted to follow up on. And when we attempted to do that, we either discovered that that was fake news and published a paper, or we discovered that it was fake news and moved on without publishing a failure to replicate. It can be difficult to establish um, why a finding is incorrect. Finding that is in the literature might be incorrect because of a general problem or a specific problem within the lab that is forming it. Like one of the things that we tried to replicate, which ended up being a letter to the journal Cell, involved PCR. In the early days of polymerase chain reaction, PCR, where you amplify a gene, thus purifying it, and then you can also have a lot of the DNA to analyze. In the early days, there were often problems with contamination leading to leading to false positives, and so that was the case for a paper. There was another paper that seemed to provide an important clue, which is by one of the leading geneticists in the field at the time, namely the former president of the American Society of Human Genetics and the editor of the American Journal of Human Genetics, David Cummings. And so in this PC1 Duarte finding, where it was supposedly a protein polymorphism linked to behavior, what we eventually found was that really the whole thing was an artifact. And subsequently, that was never replicated by anybody else. It was more or less dropped by Cummings. But, here was a paper in Science. We worked on it when I had a relatively small lab for about 6 months, and it was really a dead end. So, there are a lot of challenges that you face, but I think that especially when your lab is small and you're then in a more vulnerable position. Another event that I remember very well was when I was a medical student and I was working part-time in a molecular neuroscience lab in Galveston, Texas. This is the lab of Michael Ziegler. And I was doing assays for some of the catecholamine biosynthetic and metabolic enzymes. And in doing that, there's a crucial phase where you bind the radioactive product of the catecholamine enzyme reaction to alumina, and after doing that binding and then washing the alumina, which is powder, then you either assay the supernatant by scintillation cannon, or you assay the alumina. I think you assay the supernatant. And so as I was throwing out the non-assayable portion, and as it was hitting the bottom of the trash can, I realized I'd thrown out the wrong thing. And this is really very difficult when you have so little time. You know, I was doing all of this in my off hours and part time, and really it's your only project. And you know, I see that a lot in young investigators, graduate students, and students today, that they take so much on individual experiments. And if there should be a problem, it might be a bad reagent, it might be a bad idea, then it's really quite difficult for them. I'll just mention one other example. We, in the early days, were cloning genes, and we cloned a couple of genes. And you know, nowadays you don't do that because we have the whole genome sequenced as of 2000. So to do that, you first purify the protein, then you send the protein to another laboratory to do peptide sequencing. If you don't do the peptide sequencing yourself, which few labs do, get the peptide sequence back, and then design DNA probes to screen the DNA library. So we sent our purified protein to a university laboratory that does this on contract in California, and back came a sequence, which we looked at the sequence, that didn't really make sense because it was not like any alcohol dehydrogenase, which was the protein that we were purifying, but it was something else. And we duly screened the library, and we cloned out an HIV envelope protein because they had sent us back the wrong sequence, and they'd sent some other laboratory our sequence. And so then that other laboratory, I'm sure that they went through a similar process. So you can, in science, always be victimized, if you will, or stumbled, as you say, by these sorts of inadvertent mistakes. Sometimes your mistake, and sometimes that of somebody else.
You have told, I've heard you tell a story previously about how your genetic work corrected a misconception that had been proposed. I think you said there was an idea that heavy drinking among Native Americans was a cultural norm rather than something
Right.
biological. Can you tell us about that?
Yeah, thank you for that question. It's really an important one. When, of course, our studies in Native Americans were largely geared towards trying to identify genetic factors that were involved in, you know, they're responsible for vulnerability, either to becoming an alcoholic or consequences. But in the course of doing that, you inevitably also do some genetic epidemiology and some epidemiology, and you look at other causes. It's very dramatic, you know, that when you look at different American Indian crimes, that that there are quite different rates of alcohol use disorders. It's just, you know, it's just a misconception that they're all plagued by alcoholism. Yet the tribes that we studied, several of them, the rates are very high, and the leaders of these tribes view alcoholism as their number one problem, responsible for so much of the violence and transgenerational trauma and unemployment, and also medical diseases that are consequent. And so a lot of people have thought carefully about why the rates of alcoholism and other substance abuse can be so high in Native Americans. And it's been proposed that the diagnosis of AUD is really a reification of a culturally normative behavior. Before our work, there had been work by a group at University of Colorado, Jim Shore and Spiro Manson, where they actually psychiatrically interviewed Indians, and they found that the nature of AUD was similar in these Indians. And we found the same thing in our studies on Southwestern Indians in the Plains Indian tribe. That when you look at any of the DSM symptom areas, like problems with the law or unemployment problems or family problems, that regardless of which DSM symptom area, they're always worse off, and that the people who binge drink are even worse. And the binge drinkers are worse off even if they don't have the DSM diagnosis of AUD. So we believe that that what some cultural anthropologists had been proposing really was misleading. And probably the tribes themselves were wise to keep alcohol off their reservations if they could. If you go to Indian country and to places where there are reservations, it's really quite striking that you'll have a large reservation, then right at the border of the reservation is a Circle K store selling the fortified wine, which is basically purchased by no one except a person with alcohol use disorder. That's the only person who will drink that, because really the only purpose of drinking it is to get drunk. And so the pattern in these reservations could be that when the unemployment check or whatever money arrived, that then the person with alcohol use disorder would then hitch a ride to the edge of the reservation, get themselves the nearest place where they could buy this sort of alcohol, get blind drunk, and then sometimes walking back to the reservation actually get run over on the way back. So it's quite an interesting phenomenon that they have not learned normal patterns of drinking. And so the idea was, well, if we just provide the alcohol, then they would learn normal patterns of drinking. But the evidence is that if they have access also, as in towns in western Oklahoma where we studied Native Americans, and they can get alcohol quite readily there because they don't have reservations, but they also have very high rates of alcohol use disorder.
That's incredibly destructive.
It is incredibly destructive. The strongest prediction or the strongest predictor of alcohol use disorder is not genes. The gene, there is heritability within populations, but when you also look within populations or between populations, the strongest predictor was early life stress and trauma, and also these other correlates such as unemployment.
Something more happy. Yes. Have you experienced any what you would call eureka moments?
Thanks. It's a great question in science, and the answer would be yes. There are several times where we've had discoveries that led to papers in Science and Nature, and one of the things that I would say is most exciting is when you see that you find a functional locus, like we did at genes like neuropeptide Y and the serotonin transporter, and then you are able to trace the effects of this genetic variant through the intermediate molecular mechanisms, and then to brain intermediate phenotypes that mediate a complex behavior. So an example of that would be neuropeptide Y is an anxiolytic peptide, we found a functional polymorphism in the promoter, the control region of that gene, and then worked with a couple of the leading groups in the world on the neuroimaging the neuroimaging phenotype that should be altered if you disturb the function of this anxiolytic neuropeptide, and then looking at anxiety and pain threshold itself. So to be able to trace the effect of a gene all the way from sequence variation to molecular effect to the intermediate phenotypes of the brain, and then the final complex behavior is very exciting. That's the whole goal. But I would go back earlier in my career, because that which you find earlier in career when you're just starting is always I think the most exciting. And it would be that I came to the NIH first to do proteomics. And we were using a method called 2D gel electrophoresis, where you can look up to 1,000 proteins at a time in a single cell. And so we were doing this in order to understand the protein changes or differences that were markers or part of the mechanism of diseases like Lesch-Nyhan disease and Alzheimer's disease and other things. And as I was doing this work, that's when I really became a geneticist, because on these protein patterns, these complex patterns with 1,000 protein spots, I noticed that there were a number of these, a small number of them, that jumped in their positions. And this was because they were polymorphic with missense variants, missense amino acid substitutions that altered the charge of these proteins and caused them to move from one position to the next. And so then that was the beginning of a lot of other science where we used these genetic variants to map the proteins, looked for their linkage to other phenotypes, including behavior, and used them in evolutionary studies where we compare different species or look at the genetic variation within species. And it prepared me really to move on into the DNA sphere. And it also prepared me for a lot of what I'm doing today with transcriptomics, which is another form of omics, and where you quantitate many different indicators of molecular function. And you again have to use a lot of the exact same tools as I did with the proteome. So it was that moment that really was a eureka type moment, especially because the belief in the field was that these proteins were not genetically variant as far as their positions being altered. They were supposed to be like a common unvarying framework that you could do your quantitative work in, much like the stars in the sky. But just like the stars in the sky, there are a few of them that move. They could be planets, or they could be meteors or asteroids. And so, understanding that which is constant, but also understanding that which is varying is very important, even if it's only a small percentage that are variable. It is that small percentage that's important. This goes right to the DNA variation that we have. It's about 1 out of every 1,000 DNA bases that is genetically variant in most people, or heterozygous, as we say. So 999 out of 1,000 are not genetically variant. But that 1 in 1,000 that are variant is responsible for all of this heritability, molecular variation that people have to individuate one person from another. That's why alcohol use disorder or anemia or whatever the phenotype might be is heritable, because there's actually variation in this relatively small number of DNA bases. But, even if it's a small percentage, when you have 3 billion DNA bases, that's a lot of genetic variation.
Now I have a philosophical question I have wondered for a while. Alcohol use disorder clearly is it's destructive. There are genetic variations that go with it, and this happens for other conditions, causes variations in the population. Suppose we get to the point where we could change those to what we would think is a correction. How risky is it that those things might hold some benefit to humanity, those particular things in other contexts, and we would be giving away some potential benefit to humanity?
And it's very likely that it would boomerang. The result would be that you would make people much more of a plain vanilla type, uninteresting crowd. The genes that predispose people to alcohol use disorder are probably many of them the genes that make us neurogenetically individual, that make some of us more anxious than others, that make some of us more risk-taking. And we need our risk takers, and we need our thoughtful people also who are more cautious. And if we look at alcohol use disorder, we certainly have so much heterogeneity as far as causation that it would be a matter of eliminating or correcting one type of difference. There are some people who drink because they're anxious. There's some people who drink or they're not anxious enough. Both sorts of traits are heritable. And if you look at societies, it's very likely that the most successful societies are ones who have a balance between the risk takers and the thoughtful, more anxious people. Some of the actual polymorphisms that have been identified seem to work as balanced polymorphisms, where they have an upside and a downside. So an example would be the COMT polymorphism, which is a missense variant Val158 met of this enzyme that metabolizes catecholamine neurotransmitters. Neurotransmitters like dopamine and norepinephrine, just noradrenaline, adrenaline, and so if you look at this enzyme polymorphism, the people who have 2 copies of valine are metabolizing dopamine more rapidly. And so they tend not to be quite as acute in their executive function due to the fact that they have lower dopamine levels in frontal cortex. On the other hand, the methionine individuals like myself would tend to be more stress reactive because they have higher catecholamine levels. But under ordinary conditions, maybe they have somewhat better frontal function because they have these higher dopamine levels. Drugs like amphetamine and methylphenidate can change these frontal dopamine levels, as you know. And in most people, if you give drugs that increase dopamine levels in frontal cortex, you'll improve executive function, measured many ways, measured by, for example, perseverative errors, and measured by Trails tests, the many tests, or the famous NBAC test. So in many ways, but people had been onto the fact for a long time that you can manipulate your cognitive ability with drugs like amphetamine, which is precisely why sometimes people abuse these drugs, and they cause great problems as they continue to take them, of course. So a standard treatment of ADHD would be methylphenidate, which boosts frontal dopamine and takes a person who might be more of a val phenotype and makes them more like a met phenotype. So what should we do? Should we say that one genotype is better than the other? No, because we need both. And that's only one gene. And when you think about the polygenicity of most behaviors, the fact that there are hundreds and thousands even of genetic variants that could impact them, that the idea of making a person who is a newborn who would be suitable for all potential human environments that newborn could face is quite naive, because it could be a time of war that is approaching. We could need somebody to do the hunting, or it could be a time when we need somebody who is back in the cave, um, actually um, making the art or devising the next new invention, like the wheel. So, just to map it at the level of one gene, we can see that it would not be wise. But especially when you think about the complexity of causation of behavior, we see that even if you could engineer particular behavior, that would probably boomerang. So I think that it's a crucial question, and it will be very much more important as our tools to manipulate the genome increase. Today we have the ability via CRISPR gene editing to do precisely the sort of editing that we're talking about. Today there are ongoing clinical trials where the sickle cell anemia variant alanine 6 val is being edited to correct sickle cell anemia. Why would that have been a disaster in West Africa 150 years ago? Well, it would have been a disaster because whole populations might have been wiped out by malaria, which this polymorphism is protective against. Now, today, malaria could be treated other ways, and many people who have that genotype are in places where there is no malaria. So it makes sense to edit that particular genetic variant out. But a genetic variant like the COMT variant, you would never want to edit out. That variant has about a 50% frequency worldwide, maximizing the genetic diversity at COMT, the functional diversity at COMT. Why is that? It's very likely because worldwide, across populations, there's an advantage to having some people who have one type of neurogenetic predisposition and other people who have another. And so you maximize that by maximizing the frequency of these two alleles, and somehow that's what natural selection has done.
Very thoughtful answer. I hope that people listen to it in the future. So alcohol research is very interactive. It's highly networked now and in the past. So what people do you consider to have had an important impact on your research and on other work in the field?
Thank you. Well, some of them, you really know and you work with, and others are just people whose footsteps that you've followed in. And so, on the human research side, I, fortunate to know Bob Cloninger, to follow in the footsteps of people like Mike Bowman, and they were involved in the early studies of twins and adoptees that really laid the groundwork for the genetics of AUD, showing that alcohol use disorder and related conditions were inherited, and that you could see this heritability even if you looked at adoptees who did not know their parent. They still carried the genetic risk that their parent had. And then two of the people who are now passed over to the other side, who I would point out would be Henry Bickleiter and Ted Reich, and these were the founders of the COGA study, which I was actually involved with at the beginning, but then I went off to do my own studies in population isolates in Finland and Native Americans. And and Henry was very important in terms of really bringing the intermediate phenotype into powerful play in the alcohol addiction field. He looked at the P300 event-related potential, which is a attentional electrical signal in the brain, and showed that if you looked at the young male offspring of parents with AUD, that they already, many of them had this attentional deficit measured by P300. I encountered and was able to collaborate with Richard Dietrich, who on the animal side had done such important work to select strains that were high and low for alcohol sensitivity. And much of what they produced in terms of correlations to this alcohol sensitivity were due to random genetic fixation. That these animals, these long-sleep mice and short-sleep mice, were largely inbred. And so anything that might be genetically fixed in the LS mice would then be different than what was genetically fixed in the SS mice. And I didn't realize that, but I was able to do a study with him and with his statistical geneticists showing precisely that. And his openness, to the problem of the inbreeding of these animals into this problem was fantastic. I mean, when you think about the fact that they had published literally hundreds of papers on the LS and SS mice, and that he was able to reach out and work with really a neophyte in the field on an issue that impressed me so much. And always I saw that in Dick Dietrich. I was strongly influenced by a couple of molecular biologists, one of whom I knew well, the other one who I barely knew. The one who I barely knew was Akira Yoshida, who is a Japanese geneticist who is really responsible for the early cloning of alcohol dehydrogenase genes, and not well known in the States, but his work was profound. And then a scientist who eventually became the director of NAAA, T.K. Lee, and T.K. Lee did so much foundational work in the alcohol dehydrogenase and aldehyde dehydrogenase enzymes. In particular, was very thoughtful about what it meant to demonstrate a gene-gene interaction as opposed to a gene-gene additivity. And what he showed with these two genetic variants, which alter alcohol use disorder risk by leading to the Asian flush because of the accumulation of acetaldehyde, they're in consecutive steps of the alcohol metabolic pathway. So you might think that they would act synergistically, but he had a beautiful paper showing that if you had this genetic variant in ADH and that genetic variant in LDH2, these two genetic variants that both act to elevate acetaldehyde levels, that instead of acting synergistically, they acted additively. And his paper in which that was demonstrated was really a beautiful paper. He also influenced me because he created a rat called the alcohol preferring rat. This is one of the selected strains. And one of the pieces of work that I was happiest with that my lab did was to actually, after he had shown that there was a genetic alcohol preference by virtue of the fact that he could create this strain, people worked with the so-called P rat, the alcohol preferring rat, for many years. And shortly before TK died, we were actually able to do the genomic analysis of the alcohol preferring rat, and we didn't find all the genes that are responsible, but we found one that is definitely responsible. It's a stop codon in a metabotropic glutamate receptor 2. And so here was a functional polymorphism that actually wipes out the function of a major neurotransmitter receptor that regulates the release of glutamate by neurons. And at many levels, we were able to show that this polymorphism, this gene knockout, if you will, was one driver of alcohol preference. So people like him who worked on the alcohol preferring rat for decades, and created a tool that we could then use. I was so happy to be able to do that. I would like to also to mention that when I came to NIH, that it was a profound thing to join a lab of somebody like Carl Merrill, who was such a polymath and really a great thinker. Many times he actually came out of his office and worked side by side with me at the bench as we did our two-dimensional electrophoresis, but also to help me understand how to create antibodies to many proteins, and that we could identify what they were on the gels, or to purify proteins, so we co-electrophoresis them to measure enzyme activities. And so Carl was so creative and proficient in the lab. He inspired me a lot. And then when I did join NLA, I think I was fortunate to join the lab of Markku Lanoila. He's a Finnish scientist. He'd actually been a boy television star. His guitarist always kept his fingernails perfect so that he could pick the guitar strings. Had a minimum of 15 pins of all colors and all working properly. And what he used to do when I was a unit chief in his lab, which is the lab of clinical studies, was that he would see me in the hall or in the lab, and every day he would ask me one question about how a certain project was going until that project was brought to completion. And so it was easier it was a motivator, if you will, to complete the project, because you knew that, that Markku would be asking. He also was very important in us being able to do our studies on the Finnish population isolate. And he introduced me to Matti Virkanen, who's a Finnish psychiatrist on a forensic psychiatry unit at the University of Helsinki. We were not only able to study Finns, who represent a population isolate where these heterogeneities are reduced, but to study some of the worst early onset alcoholic patients in existence. So these were violent offenders. Many of them were actually murderers and arsonists. They were highly impulsive. They have a high frequency of another arousal syndrome called borderline personality disorder. And via studies on that population, we were able to see in strong relief or salience the effects of certain determinants of alcohol use disorder, namely the determinants that are related to arousal and impulsivity, determinants like serotonin, and genes like the serotonin 2B receptor, where years later then we found that there was a stop codon that is very common in Finns, but much more common in these particular Finns. And so that was a paper in Nature, actually, the discovery of the stop codon and the demonstration of what it did to lead to intolerance for delay and to hyperarousal. So there's so many others that that I could mention. The last I would say would be my own father, who is a pediatric immunologist, and Dad himself, before he passed away, had published somewhere between 100 and 200 papers. He had a distinguished career, but also it was just great that I was able to actually go on rounds with him. It was different days when he would make rounds on his asthmatic and often very sick kids, and to go to the lab, and he was a cellular immunologist, and to see the different sorts of analyses that he would do. It might be at the antibody level with the famous Uchterlony plates, where you can see the immunoprecipitant arcs from the different antibody antigen interactions, or it might have been at the microscope, where you could see the cells and see how they were stained and studied and photographed. So that ability to see the science, I think, was very helpful. And actually, it was due to Dad, probably, that was able to get, even as a high school student, into some of the labs that I did, to learn a lot of science and process of science from my exposures in these labs.
Oh, what a wonderful experience.
Yeah, you owe a lot to people, and it makes a big impression on me when you might be in another country, or far away, and you encounter somebody who was inspired by your father or inspired by one of these great mentors.
What do you see as historic moments advancing or understanding of functional genetics?
Yeah, there have been so many revolutions. There was the ability first to study individual proteins and DNA, and that was critical in the and so here I'm just doing a molecular part because it's functional genomics, and so it's got to be understood that a tremendous amount was learned at this single protein, single RNA level before we ever had the genomic revolution, the advent of which was really in about 2000. And so the genomic revolution let us understand that, that in the genome there are about 25,000 genes, and instead of individually cloning genes to seek to actually isolate them via their sequence in the genome, which a sequence that we would already have, there were methods that were critical. These methods include the polymerase chain reaction method, PCR, which Carrie Mullis is said to have invented, but actually it was conceived by the molecular biologist Khorana, Nobel laureate Khorana, more than a decade ago. But the ability to thermocycle a DNA polymerase enzyme, so that the enzyme would be active or inactive, was crucial. So people don't understand what it means that there's a DNA polymerase called Taq polymerase. Well, it means that there's a microorganism, Thermo aquaticus, which is a which lives in hot springs. And so you have a DNA polymerase that is active at these high temperatures and not active at the low temperatures. And so Khorana could do PCR because he didn't have this enzyme. With each cycle of DNA amplification, they would have had to add new enzyme and new reactants. But with the Taq polymerase, what one could do is just to cycle, hot, cold, hot clone with each cycle, then to melt the DNA and to begin a new cycle of DNA replication. So with each cycle that you approximately double the amount of DNA, actually it's 1.96 the amount of DNA. And so after only about 30 cycles of DNA amplification, you then have 2 to the 30th of whatever was the original target. And so that constituted both an enrichment of that target so that you would have enough to analyze by another method, like put it on a gel, but also a purification because nothing else is being destroyed. It's just that you're creating an overwhelming amount of your target. So that was very meaningful because as soon as you have the sequence of the DNA, now you don't have to clone it into a bacterium, which we were doing. We were cloning it into bacteria, and you grow up the clone that had that particular target. And when you have enough of it, you purify that DNA. Now you have the target that you want. But instead, to be able just at a test tube, to be able to amplify that DNA, that was crucial. Then when you look at DNA sequencing, that changed fundamentally. The sequencing first was done on these gels, and the original human genome sequence was done in a factory of these gels where there would be, about 96 lanes in gel, and the sequencing then the sequencers would fill the whole building. It required hundreds of people to run these sequencers. But in the second and third generation sequencers, the sequencing was then done by fundamentally different methods, which I'm not going to get into here, but methods that enabled us to sequence billions of DNA fragments at one time, and for a single operator to sequence those billions of fragments all at once. And that's really an amazing thing because it let us sequence not a few genomes in years, but sequence many genomes a week. The other technologies that were array-based included these SNP arrays, where we now genotype 1 million markers routinely on a single array, and those million markers let us do the coverage of the genome for genome-wide association studies. And then for the epigenetics, we moved forward so much. Instead of doing something called bisulfite cloning, where you would treat the DNA with bisulfite and then make clones representing the target sequence, you could determine which sites were methylated and which weren't. Instead, we run an array which now has 850,000 CpG sites on it, and you get an instant result for 850,000 sites spanning the whole genome and with hundreds of sites at an individual gene. So you have to point both to the increase in knowledge, where we have the sequence and we have the knowledge that there are these 22 million polymorphic sites, but you have to also point to the technologies that let us effectively use that knowledge.
What challenge do you see to take all these tools we have now to get these into the clinic? What challenges exist, and how do you see that we as researchers as maybe part of the way to get through those challenges, remove them?
Yeah, so there are different goals that people have with the genomics. One thing that's been very important is just to be able to help people who may have psychiatric disorders to understand that it's not all their fault. There's a volitional component for sure in why somebody develops alcohol use disorder. You don't develop alcohol use disorder if you don't use. And in fact, many people looking at their own relatives and realizing that there's alcohol use disorder in their family choose not to drink or use drugs. Our current president, Donald Trump, made exactly that decision, for example. So I think that just that information is important clinically because it helps people to understand that there is a widespread vulnerability and they may be particularly vulnerable themselves. But when we look at the more precise uses, the idea that we would be able to perform precision medicine to predict which patients would respond to which drug, or to predict accurately, not just that somebody has a family history of AUD, but that they're the one with particular vulnerability to AUD. We are far from where we need to be. The polygenic scores so far account for less than 10% of the genetic variants. So they're not very predictive for AUD. They're much more predictive for a trait like obesity, in fact, where they're even more predictive for educational attainment. So we're not at the level that we'd like to be, where we do molecular diagnosis for sickle cell anemia, for example. You either have the genetic variant of hemoglobin B or you don't. And if you don't have sickle cell anemia. We're not there at all. And so from the standpoint of precision medicine, we have far to go. We have a huge obstacle in getting there, which is that our clinical trials are not that large. And so if you're doing, and many of them don't have pharmacogenomic component. And so we don't really even have the data sets today to ask the question, which gene types predict a favorable response to 12 steps therapy or to acamprosate or naltrexone or whatever it might be. So that's a deficit. The actual nature of genetic predisposition to AUD is, of course, a complicating factor. So we have a few genes of large effect, genes like alcohol dehydrogenase, or ADH as we call it, or LDH2. These are genes of large effect, but even as genes of large effect, they only exert, a several-fold effect on vulnerability. But most of the genes that are being found are minuscule in their effects, and so they only become predictive for anything if you put them together in large polygenic constellations. That also gets at the identification of genes which are clues to new therapeutic targets. But there are more encouraged because when we do polygenic analyses, we point to pathways that are important. And these pathways themselves, in addition to individual genes, are the druggable targets, or the pathways that were intervention might be important. So an inflammatory pathway might be implicated, a dopamine signaling pathway might be implicated, a serotonin inhibitory pathway might be implicated. In fact, all of these have been implicated in AUD and constitute places where one might develop a new therapy or target a therapy. So I do, and you can see this with a lot of the papers and studies that are being published these days. We just did the exome sequencing of alcohol hepatitis, and in doing that, we pointed to decisively to pathways that are involved in lipid accumulation, but also, and this would perhaps not be surprising for alcohol hepatitis, for inflammation genes, but also some of the specific pathways that contribute to inflammation. And so, I do think that the challenges are great. You could say that with some justification that the genetics has contributed little to our clinical treatment or in prevention of AUD at this point. But I think that that with the power, especially this pathway type analysis, that we're going to see advances. The one last thing I would add to that is that, and it goes back to our discussion of functional genomics, where I left out something very important, which is that in a modern era, when we do functional genomics, also that we're able to do transcriptomics, that instead of looking at one protein at a time or one RNA at a time, we look at the whole transcriptome, at all the genes, and that we're able to do this at the single cell level, and now at the spatial single cell level. So we're mapping in the brain where the cells are that are disturbed in their function after alcohol, or to predispose to or part of the addictive process, and looking at the molecular signatures of single cells as they change function. Should have said that, but I'm glad that I had this random thought that might get spliced in somehow. Go ahead.
What interactions do you see between functional genetics research related to alcohol and other areas of alcohol research? And have you noted that any of those interactions have been key to develop new concepts?
Yes. So really, functional genetics depends upon these cross-disciplinary interactions. I was able to, in my career, work with really great scientists: Danny Weinberger, John Zubieta, Andreas Heinz, people who created methods to let us look into the brain as if through a window to these intermediate phenotypes. And this sort of neuroimaging is not something that I would have ever been able to do, either to have the resources to do the neuroimaging or to have the knowledge to do those studies appropriately. And so a scientist like John Carr, created a whole paradigm for measuring subjective responses to pain and to imaging the responses to pain in thalamic nuclei and amygdala and hippocampus, and using PET ligands, and so you have a whole science and technology around that. You have to build a whole team to be able to do that science. Danny Weinberger, as we did our studies on schizophrenia and other phenotypes, actually based a couple of three scientists in my lab. And so in these early days, we were doing the difficult part of the genetics, which was difficult at the time, even on this, just on this gene by gene basis. And on the other hand, relying on Danny and Mike Egan and others for the deep phenotypes via the neuroimaging. It was those phenotypes that showed the strongest effects of the genes. There's one, and oftentimes, though, is reaching out to people outside of the so-called AUD field, addiction field. Early on, when we were beginning our studies on Native American populations, I was able to get the help of Elliot Gershon, who was the lab chief at NIH at that time. And he taught us how to do psychiatric interviewing with the structured interviews. There's the SADS and SADS LA that we were using at the time. And, of course, now we use a different structured interview. And they're also experts in that area, people like Michael First. And so when our group sets up a new, you could call it technology like that we don't try to invent that wheel ourselves. Instead, we go to the person such as First, who has developed the instrument and who is most expert in applying it, and get them to teach us.
What would you like to see in the future for alcohol research in general, genetics in particular, and there's what you like, what are some things you expect to see?
Yes, so I'm going to deconstruct that question. So what I would like is what I would expect, actually.
Good.
What I would like is that I would like to see that that people who think of themselves as alcohol use disorder scientists think of themselves much more as scientists in whatever foundational domain it is that they're going to contribute most strongly. So they may be contributing most or thinking that they will contribute most strongly in fetal alcohol syndrome. In that case, they're a teratologist, they're a developmental biologist, or at the back end, when you're looking at the effects of FAS, there might be a neuropsychologist studying ADHD and studying the sorts of cognitive deficits that are found in ADHD patients and that are also shared in FAS patients. So what I think, and let's say that you're a geneticist who's interested in why the aldehyde dehydrogenase and ADH and common functional polymorphisms exist in the first place. Now you're an evolutionary biologist. You're studying populations. You're using tools that evolutionary biologists use things like measuring how long that it might have been since two haplotypes that are distinct today, two patterns of genes that are distinct today, or patterns of genetic markers that are distinct today. How long ago that they would have coalesced into one pattern? And that involves all of those tools of population genetics and evolutionary genetics. And at the same time, you would want to see that people continue to have their real focus on the tragedy of AUD and the importance of bringing these tools together against a disease that we need to prevent and cure. And I think that we should be thinking in terms of cure. That it is wrong for us to think only of harm reduction. That harm reduction is an easy way out, if you will, and it too often doesn't work. That what we should be trying to do is to actually address especially the damage that's occurred to the brain. That one of the great tragedies of AUD is that we start out with whatever personality that we have. We may be a risk taker, we may be relatively more anxious and taking this more difficult. Maybe we suffer an early life stress, and so all of this can predispose us. But our early experiences and our genome don't account for what addicted patients become. The addicted brain needs we need to actually recover it. We need to undo this. So I'm not happy with solutions to opioid use disorder to maintain people on an opioid agonist for the rest of their lives. What I want to do, and what I think the field should do, is to actually help our patients recover. Much as a patient with cancer, our goal is not just to contain the cancer, so instead of dying in 1 year, they'll die in 2 years or 3 years. But the ultimate goal is to actually restore the addicted brain. And this I actually wrote many years ago in an editorial in New England Journal on restoring the addicted brain. And I think that we will move to that, and we'll be able to move to that partly because we understand so much more about the neurocircuitry and in cellular molecular changes in addiction. And as you learn about the neurocircuitry, then you learn, as our neuroscience colleagues are teaching us, that there are these ensembles of cells that are maintaining these behaviors. Change the function of these ensembles of cells, and you will then change the behavior. And we need to do that in a way that, as much as possible, brings the person back to their pre-morbid state, to the best of themselves, instead of this thing that they become because of addiction.
For for researchers at the start of their careers, do you have any advice to offer? And given your clinical training, do you have any specific advice for physicians who want to pursue research?
Great question. So what I noticed when I was early in my career is that when I would solicit advice like this, is that much of the advice that I would get would be based on what had happened 20 years before, or even more. And it would be sincere advice and oftentimes good advice, but I think it needed to be carefully examined. What I believe is that with each new generation, there are now new possibilities opened by, first of all, the dynamic of science, the structure of science, but also the tools that we have. So, you and I, when we were postdoctoral fellows and we needed information, probably went to the library. We consulted Index Medicus and the citation index. We went to the stacks, found the journal, copied the article, and then, and it's this very slow process, accumulated information. Maybe we had a whole series of textbooks. We had many, and we would look it up in a textbook. And this process was slow, but worse than slow is that it was not comprehensive. And not only was it not comprehensive at the time, but if you compare it to the wealth of knowledge that we have today, completely inadequate. So today, if you do the next advance, of course, was that we had PubMed. And so instead of going to Index Medicus, you would go to PubMed. You would find a host of papers and you would find citations. Then there became the availability of electronic versions of these papers on PubMed. That didn't happen very long ago. And when it did, it was an enormous advantage. And it was an enormous advantage to people who had access to the electronic versions. And so it points to, you need to be in an environment where whatever the most advanced technology is for access to knowledge, you need to be in that environment. Now we moved to the age of Google. And so in the age of Google, then rather than use PubMed, very often I will Google the issue, whatever it is initially, and then the top papers amazingly come up. And that was important because if you do a PubMed search and there are tens of thousands of papers, it's really not very much help. How do you, amongst those 20 or 30,000 papers, identify the one that you really need to target? And so a Google search would often do that. The LLMs, the large language models, are now the way to go. Many times a day, I'm using LLMs. Oftentimes, when I'm meeting with a postdoctoral fellow and we're exchanging ideas, and we're trying to figure out, where we are, what something means, where we go next. And so the LLMs are an enormous help in interpreting that and in triggering new ideas. And of course, then what you have to do is that you put a little bit of trust in that information, but you must verify it and you must further perfect your questions to the LLM, or you're not using them well. So I would say that for any scientist today, that they shouldn't be thinking in terms of doing things the way that we did 30 years ago, but doing things the way that with all the power and all the tools that they have available now, which eclipse and dwarf the tools that we had when we were of about that age. They need to put, though, a lot of trust and reliance on themselves. They increasingly, science is this group process with large groups. And I think that it can be stultifying. that it's important for any young scientist to have this very strong sense of their own individuality and self, and a little bit of hubris, and the idea that it just might be better than the next person, and they can't all be. So if you go to the poster session of a meeting like this, well, you see about 1,500 people there, and a few of them will really make critical contributions to the field. Most of them will not, but it's vital for a junior scientist to have this strong personal belief in themselves, and in having that to try to measure up to it, because I think it helps greatly to hold yourself to this higher standard. I'm always very impressed when I see a junior scientist. They come to my lab, and some are quiet, and some force themselves to ask questions at our meetings, at our lab meetings, and at other larger meetings also. And I think that that's great. They're doing that because they're putting themselves on the line, and they ask better and better questions as a result. And a lot of our ability to do science these days is predicated on our ability to ask good questions. If you look at the questions that are asked, what you see is, for any field, there's a lot of sameness. So somebody does something, then a bunch of other people inevitably do the same thing. And so you have an idea that there are a lot of people who are just working along parallel lines. I think that they should preserve their individuality as scientists. They should be thinking about that which is offbeat and somebody else isn't doing yet. They should realize that there are these 25,000 genes, and that the odds that they could just choose one gene and that's the right one is remote. On the other hand, now we have omic methods. This is true in neuroimaging. It's true in our DNA studies and our RNA studies. We have these omic methods that can identify the best targets and the best individual targets and the best target pathways. So they need to be facile in that. I would say that in this new generation, that skill in using tools like R for, if you will, taking advantage of modular protein programs and using LLMs to guide creation of new programs is really crucial, because they need to be working, the young scientists need to be working with big data, at least at a point, to know which directions to go and to have a comfort with big data. But also, somehow they need to find their own niche, and their own, something that they can put their own signature on.
For researchers in general, is there anything you ask of us?
What I would ask of researchers, I think that keeping an open mind is probably the most important thing that researchers should do. I think that researchers shouldn't just follow the crowd. It's very important that researchers are critical. I love seeing reviews, as I often do, which really go into not just the strengths of the work that's in front of them, but also the weakness, and to sometimes make suggestions as to how to do it better. There can be in science a lot of tendency to follow on to a concept which seems attractive. I mean, a very good example would be adolescent drinking. Everybody knows that adolescent drinking is bad. Of course it's bad. If you are using drugs as an adolescent. It takes away from your studies. It could change the brain. It could do everything else. So you know it's bad. And the Institute spends millions of dollars on this. And many scientists study it. But it's much less well understood that people who use drugs early, including alcohol, are different than those who don't. And the twin studies from Australia and Virginia have shown that very well. And I put it in textbooks. So about 2/3, 3/4 of the bad effects of this early drinking are not due to early drinking, but due to the fact that the person who is drinking early is different than the person who isn't. And that's been learned by looking at discordant SID pairs and twin pairs, where one twin drinks early, the second twin has the same risk of drinking early, but didn't drink early. But they have the same risk because they're carrying the same genes. So there are a lot of things that people think are true and that follow along, and they don't think about these in a very careful way. And some of the ideas that people come up with are not necessarily good. They could be nutty ideas, but it's good that they're coming up with them. An example of that, I guess, that just randomly comes to my brain is a camper safe, which a few years ago, I think it's Marcus Heilig who just received an award at this meeting. And I'm blocking, oh yes, it's Rainer Spinagel, a German addictions scientist, proposed that a camper safe wasn't working by virtue of its effect on glutamate or other neurotransmitters, but as a carrier to bring calcium into the cell. So they proposed that it was a carrier because I can't remember the name of the molecule at this moment. It might come at some point, but it's a calcium something or other. And so they proposed that it might be bringing calcium into the cell and thereby modulating cellular function. Okay, that's not true, but it's original. And it is worth thinking about and exploring. There are a lot of ideas like that that are not necessarily true, but that are important for the field to be explored, because we should be exploring a lot of ideas, and everybody should not be doing the same thing.
That's it. What seems nutty one moment sometimes remains nutty, and sometimes it's a Nobel Prize.
Sometimes it is. For example, when Barbara McClintock observed the action of the transposon jumping gene, she was already well known, and she suppressed these observations that she made in the maize corn, showing transposons where all colors or all the kernels are various colors due to the actions of transposons mutating the color genes. She suppressed that for over 20 years because she knew that she would be met, and she was, with massive skepticism. Today, we know that transposons are critical. When Wegener put forward the theory of continental drift in the early part of the 20th century, he was mocked. The most famous biogeographer of the time, William Gaylord Simpson, organized conferences to denigrate him. That went on for decades. It was only well after his death, and when the striping of the magnetism of the rocks on each side of the mid-ocean ridge was observed, it was observed that what any child could observe, that the continents kind of fit together as a puzzle, and which Wegener had emphasized. It was then accepted that the continents could move. But who would think that something as massive as a continent could migrate several centimeters a year across the surface of the Earth? Nobody's sane. Nobody's saying, but there was Wegener out there alone with the continental drift hypothesis, which, as I say, during his lifetime was never accepted. He was already a famous geophysicist, and so was it, a prominent professor, and otherwise, and otherwise very credible. But when he put forward the theory of continental drift, it was met with derision.
Well, on the flip side, if he had been wrong, it would be important to know that. I'm thinking of occasionally I encounter someone who does an experiment, a hypothesis is not confirmed, it's rejected, and they say the experiment failed. It didn't. They learned something. So I think the acamprosate and the calcium, that was not true, and we learned something.
That's right. Very often, though, we're concerned with replication, and we should be concerned with replication, but we should also be concerned with verification. There are many experiments that are difficult to do, and somebody who's not very good at the technology might not be able to replicate it. This is coming from somebody who, as I say, went through many types of much of his career was devoted to trying to replicate things that couldn't replicate. But you may not be able to replicate it because you're not competent to do it, because of different population, because of many other factors. But verification is another issue. Verification is the idea that if you've made a discovery, that it also should be predictive of other sorts of phenomena, much as the theory of relativity was predictive of a variety of things before, in a testable fashion. The bending of the light of galaxies was in fact observed. The slowing of clocks, of a spaceship in orbit around the Earth was observed. So many of these, much of our science should more emphasize verification. And if you look at papers published in top journals today, that often is the case. If you want to publish in a journal like Nature Neuroscience or Neuron or Cell, then very often it's not just an issue of having a study with a statistic, but having an animal model that replicates, or a cell model that replicates or tests now the hypothesis that you developed, a different system than that which you just simply did. And it's a lot of our science today that emphasizes too much this first level of discovery. Like, we found this gene for such and such. Okay, so now what is your prediction? You've done this, but there are further steps to be taken. Is that type of multi-level science that includes verification that I think is most powerful. And it takes us forward also to the next levels that we need to move. In genetics, we know what we need to do. We need to go from our maps, which are getting increasingly good with hundreds of genes mapped, to the mechanisms, the second M, if you will, where we understand the functional locus and how that locus works, and then to medicine. We need to go M to M to M. And too often, we're just at that first level where we have the maps and we have the polygenic scores, but we've not taken it to the levels of mechanism and medicine we really need to go.
Maps, mechanism, medicine.
M to M2.
For members of the public who may watch you on this interview, do you have anything that you would like to say directly to them?
Yeah, I think that, if they look at the work of scientists, they're seeing people who are doing things that are incredibly advanced technically, and you're often, talking about people who are wonderful puzzle solvers, or at least think of themselves as such, and who are fascinated by the process of science and wanting to advance basic knowledge. But it's also important to know that there's a depth of feeling and concern about the diseases that we're studying, especially if it's a common disease such as alcohol use disorder, where we've all seen it in our personal lives, even if we've not seen it as physician scientists like myself have seen it, even if we've not seen it clinically. So, I do rounds on our clinical unit a couple of times a week, and see the carnage that alcohol use disorder causes, and you're seeing people whose lives have been damaged and who've damaged the lives of others and who know it. And many of them, one of the biggest obstacles to the getting better is, a sense of shame, a sense of wasted years. And you try to help them to understand that they're starting fresh each day, each new day, and we're trying to help them to start fresh and to improve. And so the long-term work that we're doing in the labs and with our studies are really aimed to create knowledge that will be foundational in making those advances and understanding that the reason why we have the drugs that we do that are of some help, drugs like acamprosate and naltrexone and disulfiram for alcohol use disorder. The reason why is because there were people who developed the foundational knowledge in labs. We had to learn what the opioid receptor was, and naltrexone was an antagonist at that receptor. And so that was the work of a behavioral pharmacologist going back actually for an amazingly long amount of time with many false leads, but also successes. And so it takes people who are really curious and dedicated and good at solving puzzles to do that work. But you should know that many of us keep very much in mind the daily suffering of people and families and communities that have these diseases. And many of us may not have the disease, but we have something. When I did our studies on Native Americans, one of the things is that on a yearly basis, I would need to meet with tribal councils. And many of the people on tribal councils were themselves recovering alcoholics. And I remember that the vice chair of one of the tribes asked me if I myself had alcohol use disorder. And I said that I didn't. But on the other hand, that that I had a sincere and strong appreciation for what AUD was, and that I studied that there were things wrong with me, and on the other hand, that with all the things that I study as a psychiatric geneticist, it was good that I didn't have all of them. I couldn't have all these things, but and both as a young man, but also as you get older, you may in your own life, but in the life of your family, encounter disease and disability. And I think that it's very important in giving you, even if you're not a physician, an appreciation of why it is so important that we help people with AUD and other addictions.
Thank you, David.
You're welcome.